We hope all of you are really excited for today's session. And we promise that it's going to be really, really fun. It's going to be really different kind of TTS session. We've not done something like this before.
Hi, hello everyone. Hi, I am Nizam Sheikh and I work with Dr. Karishma Kaushik, whom you know, by the way. She is one of the people responsible for Talk to a Scientist. All right. So this is one of its own kind sort of experience because we all hear about like Talk to a Scientist is all about scientists speaking about their work, what they do in a lab, their interpretations. But have you ever wondered what one goes through in a day-to-day life, what a scientist does in a day-to-day life? Now, I speak as a capacity of a microbiologist, and I'm going to give you a little sneak peek of what I do generally, with an exception that there would be bunch of excited and curious kids watching me do my job, which I normally do. And I'm really excited to show you all what I do exactly. Now, apart from being a researcher, I am also sort of a movie buff. And, I have also collated these images that you could see. There is Jurassic World, Avatar, and there is Black Panther, three of my very favorite movies, which shows laboratories in general, right? And these are unrealistic laboratories, by the way. Now, if you do see, uh, we have DNA popping up in the air, flaring up in the air, and some scientist who is either a lead actor or a villain, they would just punch in some keys on the keyboard and then just swap DNA or gene. This is not how laboratories are. I hope it is that way in the future. Our jobs will be easier, but not yet. So what I'm going to do is I'm going to give you a picture of what people do in a laboratory, in a microbiology laboratory. Now, before I get started, I want to make the rules very clear for today's movie. Now, it's very simple. The main lead for today is microorganisms or microbes or bugs or organisms, whatever you want to call them. Now, we are well aware of the fact that our environment around, it's not sterile, it's not free of microorganisms. We have microorganisms all over the place. It has not, yes, Tony Stark's lab, yes, absolutely, yes, that's exciting. So we have microorganisms everywhere. We have something called the microflora, which means the environment is not sterile. It is full of microorganisms. So in order to study microorganisms that is of my interest or anyone's interest for that matter, we need to work in confined spaces, enclosed spaces, spaces which are free of microorganisms. And I'm going to talk more about that. So remember this word, that whenever I want to, or any researcher in a microbiology lab wants to work, that person will have to work in a confined space, closed space that is sterile. And whatever that person is using, mostly it should be sterile, or to say the least, disinfected. Can we please move to the next slide, please? All right. Okay. So any movie comes with disclaimer, right? Now I have an activity for you. Can you all, I'm going to talk about safety, and let's talk about do's and don'ts over here. Now, can you all look at the pictures and can you elaborate a bit on, I've put it out there and I'm going to do all of the things that as I'm speaking, of course, but can you elaborate a bit on, say, for example, if I say no drinks or no food, why do I say that? Just type in, or if I need a lab coat or gloves or closed shoes, why do I need that? Can you put in one or two words of why do you think it is so?
Okay, great question. So let's maybe start with the food. Why don't we want food or water, or why shouldn't we just have a nice, our favorite juice next to us while we're working in the lab? Does anybody want to type in what they think? Why can't we have all of these favorite things inside a lab? Bhavik says
Yes,
it could lead to
absolutely. Very
contamination.
true. Chemicals, absolutely. Someone said dangerous, right? Someone also said dangerous. Yeah. We don't want to ingest, uh, be it chemicals or be it microorganisms. We don't want anything near our body. So we need to have protective layers on our body. So I'm going to, like, I have a wristwatch. I'm going to remove it because right after this, I'm going to work in a lab in the hood. So I'm going to remove this. I also have my lab coat with me because we always have lab coats around. So I'm going to put this on, right? And I'll put the gloves whenever I am going to work with anything, because whenever I'm handling a chemical or I'm handling microorganisms, because I'm also going to use the laptop at some point of time. So I don't want to use gloves anywhere but my chemicals or microbes for that matter. So yes, you are right. So we don't know, apart from microorganisms being hazardous, and we do not know what hazard level it belongs to, what it would do to our uh, to our system. We also work with chemicals which are irritants, which are flammable, which are corrosive, which is why we need to take all of these practices. And I promise I'm wearing a closed shoes. I can't show it to you right now, but I promise I'm wearing one. I have a lab coat, I have my gloves with me, and I'm really excited. All right, can we move to the next slide, please? Now I'm not going to over elaborate on these equipments. The reason is because you all have come to watch me work and not speak the whole time. I'm going to give it 2-3 liners right now. And whenever I do shift to a certain equipment, I will elaborate more on it, and I will talk about why exactly is it important, right? So let's start with the main equipment that is not the main, but it's one of the main equipments, that is a biosafety hood. Now, biosafety hood, it enables a sterile or micro-free environment for me or any microbiologist to work in. And how does it do that? How does it create a sterile environment? We'll see it in some time. Apart from that, we also have freezers and refrigerators. Now, what happens is when we want to use a certain microbe or grow a certain microbe, we revive it or resuscitate it from something called as microbial stocks, right? Now, these microbial stocks, they are stored at sub-zero temperature. And by that, I mean minus 80 degrees or so. Now, why is that? Because at that temperature, the metabolic activity will be very slow, it will be dormant, but it will not die off. Right. So whenever I want to do an experiment with a microbe, I will directly take the microbial stock. I will, grow it in a nutrient medium containing, nutrients for the organisms to grow. And also, you might wonder that, for example, microbes are grown in sub-zero temperatures, minus 80 degrees. It's very lethal for the microbes, mainly because there are ice crystal formation, right? But there is also something called as cryoprotectant that will protect the microorganisms from any harsh effect of cold.
Okay, so you mean they're stored at sub-zero temperatures?
They are stored.
They're grown at.
They're grown at a normal temperature, but they're stored at. So whenever I want to, take it out, whenever I want to grow a microorganism, I will take them out and grow them in a nutrient plate and incubate it at a certain temperature that I'm going to come to now.
Sure.
And refrigerators, certain chemicals are very sensitive to heat, which is why we store them in refrigeration conditions. Now, incubators. I'm sure you all have heard of incubators in hospitals, right? Now, incubators over there exist to give the best environment, right? And so goes over here. In our case, incubator will give the best environment for our microbes to grow. Just giving an example. Now, our body temperature is 37 degrees Celsius, right? Now, any microbe that has to infect human beings will have to have the optimum temperature at 37 degrees Celsius, and they grow happily in 37 degrees Celsius. Now, I cannot, grow them in certain nutrients and just put it anywhere. I need to put it in an incubator where the optimum temperature or the best temperature is given to the microorganism.
Sounds good.
Right?
Nizam, are we going to see any of these equipments now?
We sure are going to do that. Yeah.
Why don't we do that? We can.
We can do that. So I'll switch to my phone, and whenever there is a tool, we are going to see that. I'm going to elaborate more on that.
Sure.
Right. And if we need more oxygenation or more, shaking conditions, even that is enabled by an incubator. Can we move to the next slide? Yeah. Of course, if you're working in a observing microorganisms, we need to, because they are micrometers in size, we need to have a microscope which is containing a bunch of lenses and, making observation very feasible. And apart from that, as I said, we need sterile conditions. So whatever food that goes into it, or whatever food microbes use, should be sterile. Whatever pipettes or tips or plasticwares we use should be sterile. And I'll speak about the analogy of autoclave to a certain kitchen appliance very soon whenever I show you one autoclave. All right, can we move to the next slide? Okay, now we are moving to chemicals. So, okay, I have a tray with me, and I promise I show it to you. But now we spoke about where organisms are going to, where are we going to grow the organisms, right? Now they would also need food, right? We all need food. Now, microorganisms, we grow microorganisms in something called as nutrient broths or nutrient plates. Now, this is an example of nutrient plate. I don't know whether you can see it. We'll prepare these, but these are solidified. These are solidified plates. So on the surface, we grow the microorganisms. Now, these are basically chunks of proteins, carbohydrates, salt for the organisms to allow to grow. And this is a liquid medium. Can you see the liquid shaking?
Yes.
It's yellow in color.
Yes.
Right. So we also grow in that. It's basically food for the microorganisms. It comes in different names, but it's the food for microorganisms. And we'll talk more on this food a little later. And we have certain chemicals, stains to visualize the microorganisms. Now, shall we move to questions? Can we have some questions if you have any questions?
Let's actually see them first. Can we take a little tour before we get more questions?
For sure. Okay. Okay, I'm going to switch on my video actually in that case. Right. Okay. Or okay, Snehal, you might have to, because the slides is on, which is why.
Yes. So what we're going to do now is I'm going to stop sharing the slides so we can all see Nizam's video.
Yeah.
Great.
Can you?
Yes.
Can you see it?
Yes.
Yes. We can.
Okay, so we are walking inside a lab, Nizam. Is that what's happening?
Yeah. Yes. Absolutely.
Okay.
Now, this is the incubator that I was speaking about. I am going to switch this on.
It's rather large.
Right? Okay. It is very much large. And
oh, wow.
You can see something shaking, right?
Mm-hmm.
Yes.
Can you see? Okay, I'm going to open this. Now, this is where the microorganisms, these flasks are where the microorganisms grow. Right? And
so those are like the liquid media, the liquid growth.
The liquid growth medium, yes, absolutely. We also have the biosafety cabinet. I'm going to work in one in a moment, and I'm going to speak more about the biosafety cabinet. But this is exactly what a biosafety cabinet looks like. And Nizam, apart from that,
just hold it on the equipment for a few minutes so they observe all the parts.
Yes.
Ask them to make some list of their observations. What all did they see as we go through the session?
Okay, yeah. I'll just go to the next one because I can switch on the light actually.
Sure.
The incubator, in the safety incubator where we are going to work.
Okay.
All right. So this is what you can see.
This is again like a biosafety cabinet where you're going to work.
Yeah, absolutely. So I've arranged all my stuff, all my plates, my tubes, my rack, and everything in the hood, and we're working in something.
Okay.
Can we also do a freezer or a fridge?
Yes. Yeah. Okay. So this is your normal. You might feel that it's a normal kitchen refrigerator, but then this is actually 4 degrees Celsius, 4 to 6 degrees Celsius. Where can you see the chemicals here? There are a lot of chemicals here.
We can see
lots of chemicals, right? Right. Okay. Great. And this is a minus 20 degrees.
Okay. Minus. That is 4 degrees and minus 20 degrees.
Minus 20 degrees. Yes.
Oh, that's cold. And we also have minus 80 degrees, but that must be in another
downstairs actually. Yeah.
Nizam, Anirudh asked, why was TBB written there?
Okay, I will for sure I will answer these questions actually because I'm going to work with one. I'm going to also show you the static incubator.
Okay.
So can you see this huge
Yes.
cabin,
Oh,
right? Can you
Yes.
see the
yes.
temperature here? Can you see the temperature here?
Let them
Yes.
answer it, Nizam. Let them put it in the chat window.
Yeah.
What is the
Yes.
temperature there?
Okay. Can all of you type in what temperature you saw? Yes, Avishai says
Very good. How
36.9.
observant is that? We have all laboratory auditors also now on our Zoom. Huh.
That's amazing. That's great. All right.
Right. So that was 36.9. So it's almost close to 37, right? Which is
37.
the
Absolutely great.
temperature at which our body exists and so many microbes grow. Um, so obviously, because all of you must have seen, even your fridge at home sometimes fluctuates. Sometimes, if you have an oven at home, even the temperature for that might fluctuate a little. So it's the same thing, where 36.9 is almost 37.
Right. So, Sneha, shall we
All
do a
right.
little recap of the three equipment they saw? Maybe they can write the name in the chat window.
Yes,
Quick.
exactly.
What were the three equipment we saw in the lab? Three big, large equipment.
Yes. So the first one is one in which, yes, Avisha has already answered it before I could give any description. Yes, an incubator was the first one. Fridge, yes, we saw fridge, freezer, and we saw biosafety
Very
cabinets,
good.
right?
Yeah. Yeah.
Or
Yeah.
hood.
Great.
Perfect.
Amazing. Amazing. Amazing.
That is perfect.
Yeah.
Okay, now, Nizam, shall we look at some? What do you want to do next? You want to go back to the.
So yes, I want to go back to the slides because I just have to show them certain plasticwares and liquid handling techniques, and then we'll
Yes.
move right into uh, yeah.
Okay.
All right, great. That's amazing. So we have seen, just to recap, this is a liquid medium and this is a solid medium, and we'll talk more about it. It's basically food for microorganisms. Now, the second part is we have, certain plasticwares and glasswares that we work with. Now, these are tubes. If you can see them, these are basically tubes. And petri dishes that we spoke about. I'm not opening it because these are sterile. As I was speaking, these are sterile. These need to be opened only in the hood. So these are basically petri dishes where I put my solidified medium in. Okay, all right. And these are certain well plates, basically different kinds of well plates, where each well is basically an experiment on its own, right? For example, if I want to, grow my organism in presence of sucrose or glucose, and I want to test which one is better, I need not use different types of tubes. I would use these. These are 96 wells in itself, and I can observe them. So it's sort of miniaturized form of what we normally do. And we have certain beakers, certain containers where we would put our medium or chemicals in. So this is all about plasticware or glassware. Now
We
comes
can
the
also call them consumables, right? Because we basically consume so much of them. We need so much.
very true. Absolutely. So exactly. So which is exactly what I want to speak about. That we did speak about equipments, but also there are a lot more that goes into the game. It's also plasticwares and glasswares. Basically, we consume so much, so it's basically called as consumables. And the very next part, can you please, yeah, can you show an experiment, sir? Very, very soon. I get your excitement. I very much get your excitement very soon. Just this last bit, I'm going to do an experiment for you all. All right. So, liquid handling. Now, I have, okay, no problem. Okay. So, we work with different kinds of pipettes. If you can see, we have pipettes, micro pipettes. Now, I have a very small experiment for you, or an activity in your head. Now, uh, this is a bottle. It's not half filled. Actually, it's around 300 ml. Now, if you divide this, or you divide a 500 ml bottle in your head, which is filled with water, and you divide the contents in the water into 500 different parts, right? What do you get? You get 1 ml for each part. Now, when you divide this into more thousand parts, you will get 1 microliter for each part. That is the extent of liquids that we handle. So we have a pipette, This is 1000. I'm sure you would not be able to see it, but I believe me, it's 1000 ml microliter. This is 20 microliter, and we dispense liquid using something called as tips, right? These are basically tips. You can see it on the screen as well, and I'm going to show it to you, and we'll see this more of this. And for wells, we use something called as multi-channel pipettes. Many channels, many outlets for tips, and we can work it that way. Now, enough of me speaking, and let's get started with the experiment now. All right.
So Nizam, we saw
Just a
large
minute.
equipment, we saw small equipment like microscope and autoclave, not live, but on the slides. Then we
On
saw
the slide, yes.
chemicals, reagents. Then we saw
Yes.
plastic materials, and then we
Yes.
saw small tools like pipe
Tools, yeah.
that we use.
Right. Exactly.
So now we are going to
Exactly.
do 3 sets of activities that use all of this. Okay.
All of this.
Yeah.
Exactly. I'm correct.
Yeah.
Yeah. All right. Okay. So, is it medium rare? Okay. So, I'm going to do 3 sets of activities, and let's talk while we do it, and let's not present it anymore. Let's switch the camera, and I'll be doing the speaking at this point of time.
Nizam, I think we should show the next slide that shows what the 3 activities.
Yeah. Okay. Okay. No problem at all. Yeah. So these are the 3 activities that I'm going to do. I'm going to pour agar. I'm going to make agar plates. I can see Ananya, you're very excited. Okay, we are going to pour agar plates at this point of time. Then we'll make bacterial streaks on the agar plates, and then we'll set up liquid cultures.
On the
and on the agar plate, and grow bacteria in the liquid cultures. And I'm going to speak as and when I do these experiments. And like a cookery show, it's everything's going to be ready for you. So it takes time, but then I also have the ready version of it so that you all know what it looks like, basically.
So sounds good. So the first activity, Nizam, is going to be pouring agar.
Pouring of plates. Yes.
Next slide that just gives an overview.
Yes, please. Let's do that. All right. Okay. That's great. So what I am going to use is something called as brain heart infusion medium, and it's exactly what the name suggests. It's basically an infusion of brain and heart of bovine origin that is used as a medium for microorganisms. Now, this was already autoclaved. As I said, it's going to be like a cookery show. Everything's ready for us, and it's already autoclaved. And now I'm going to pour some plates. Now, some of you spoke about agar, right? Now, this has a very special property. Agar is something like gelatin. I'm sure you might have had custard, like puddings and stuff like that. Now, these solidify under lower temperatures or your normal temperatures, and they liquefy under higher temperatures. Your mom might be boiling milk, and they are still liquid. And then when she cools it down, it becomes solid in nature. This is exactly what agar does. It basically solidifies your medium. Whatever it goes into, it just solidifies it. And what we are trying to do is we are solidifying medium. So they are going to grow on the surface of growth medium. Yes.
The brain heart infusion, which is cow brain, cow heart, rich in protein, provides nutrients.
Exactly.
Agar makes the nutrients into a solid form.
Solid form. Yes, absolutely.
We need high temperatures to make it sterile. And then pour it on a plate, and as it cools, it becomes solid.
Solid. Absolutely. You got it right. You got it absolutely right.
Whole process. Are we going to watch this sort of?
We certainly are.
But okay. Great.
Yeah, we certainly are going to watch it.
Yeah. So go ahead. And by the time we'll answer questions in the chat window.
Absolutely. That's great. Okay. I'm going to switch off this camera video. All right. So we have Charvari, who is actually our camera person. So please say hello to her. She's going to help me out with a lot of these video shooting.
All right. Charvari, thank you for your help.
Thank you for your help. She can't. All right. You can come here. Hello everyone. Hi. Okay. So I was talking about biosafety cabinets, right? Now you can see something, blue light, right? This is basically UV radiation. It basically kills microorganisms. And apart from that, there is also filters above called as HEPA filters, which will, sort of trap your particulate matters. And there will be a constant flow of air, a uniform flow, so that continuously the air goes outside and there is no insertion of any uh, foreign particle or foreign material, including microorganisms. Now I'm going to wear my gloves because okay. So I'm wearing my gloves now. Right. Okay. No problem. Can you see me now?
Yes,
Can you see my
yes,
hands?
we can.
Okay, that's amazing. All right. Can you see this? This is liquid in nature. This is BHI
Yes,
agar, brain heart infusion agar, and this is liquid in nature, isn't it?
yes,
Okay.
definitely.
All right. So the first job that I'm going to do is I'm going to label my plates because that is the
Very important.
responsible way of working. So it's BHI agar.
So, Nizam, just to make them understand, you've put the nutrients and you have put the agar, or it came
Yes,
together, and you've autoclaved it.
exactly,
Yeah.
exactly.
That liquid has come out of the autoclave, and it must be pretty warm to touch, right? Because it was at a high temperature.
It is very much hot, which is why I will have to use these rubber gloves basically because
Oh,
it's very much hot. I really cannot touch using my bare hands, so I will have
it's
to use these
like,
rubber
have you
gloves.
ever taken a hot cake out of the oven? Like, I hope we don't go with it
Yes.
with bare hands. It's sort of like those gloves, kitchen mitt.
Right. Yeah, it's
Lab
kitchen
mitt.
mitts. Yeah, absolutely. It's lab mitts basically.
Yes.
All right, amazing. So I'm going to. These are our petri dishes, and we saw that this is liquid in nature, right?
Hmm.
Now I am going to. I'm opening it, and I'm going to pour it in these plates. I'm going to open the lid. I'm going to pour it like this.
Okay. So I think as soon as you poured it, like I don't know if everybody noticed that you can now see like a layer of sort of steam on top of the lid, right? That really
Can you
shows
see?
that it was hot.
I can. You can see it again. Can you see it?
Yes, as soon as you're pouring it. Yep.
That's amazing.
And Nizam,
Right.
that's some really deft working with your hands. I think I have lost this skill set. I can work with my hands when it's a keyboard.
It's it's a bread and butter. We are used to this
Yes.
at this point of time. We're very much used to it. So you know what? I'm going to pour like 4 plates. Right. Okay. So and also I'm going to store
Okay.
some of this so that later on by the end of the session we'll get to see the magic behind it. It will disclaimer. It will just solidify nothing else. So it's
Okay.
still liquid, isn't it? All
Yes.
right. So we have poured some plates. Now
So
explaining it
Nizam,
once.
now that you've poured these plates, can you tell us like how long does it take for this to become solid?
So it will take around 5 minutes to become solidified. Not even 5 minutes for that matter. It takes less than that, depending on what the temperature is. So I will keep it for around 3 to 5 minutes at this point of time.
Okay.
All right. So just a basic brush up on this. It's basically nutrients for microorganisms, just in a solidified manner. That is, agar solidifies the nutrient. And now the next thing that I'm going to do is I'm going to grow microbes on the solidified plate. So once it is solidified, I also have some of the solidified plates. As I said, it's going to be like a cookery show. Everything's ready for us, and we have plenty of plates. Just a minute. I'm just coming with the plates.
All right. So while we're waiting for Nizam to get some plates, maybe we can try and answer some questions in the chat window. So Anushka had a question about how agar is prepared. So most of the time, you can actually buy agar from companies. There are companies that make all of these biological and chemical consumables for labs, for research labs. So you can buy this agar. It comes in a powdered form, and they tell you how much you need to put in water so that you get 500 ml of media, right? They give you a ready
Yeah.
recipe.
Also, to talk about the oil.
Cake mixes, Snehan, that you get.
Yes, exactly. The cake, like ready cake mixes.
Yeah,
You get.
absolutely.
It's like that.
Yeah, we make a lot of food analogies, as you see on this channel.
Yes.
It's a microbiologist kitchen, to be very honest.
Yes, exactly.
All right.
Arav has a great question. Arav asks, does agar smell? So,
Oh,
if you ask a non-microbiologist, they will say it smells really bad. But if you ask a
yeah.
microbiologist, they love the smell of agar.
Yeah. It's like the
But
warm smell of, I don't know, protein.
Yeah, it does. It does. It
What a
certainly
warm
does,
smell of
actually.
protein. What is that description?
there's no way to describe it. You just have to come to a microbiology lab and smell it.
All
Um,
right. So, you know what I'm going to do right now? I'm going to put these plates at the side and I'll get to the next task that we have at hand.
while you're doing
It's
that, we take one more question, Nizam. What is the device next to the green place? What is
Are
the,
you
Uh,
talking about this?
so the
yeah.
green thing is a
Oh, that
stand.
so this is a burner basically. So as
Okay.
I spoke about sterile environment, if you want to add an additional layer of, you can burn it. This is a burner basically. You can burn it and you can work around this area. It sort of creates a septic or sterile
So it's a
environment around
flame.
the burner.
It's
It's a
like a
flame,
flame, like
yes.
a
And I
candle.
will
But
light it for you all.
Let us know, Avishai, if that's what you were talking about. But there are lots of stuff there, so you can
yeah,
ask anytime. Yeah.
Yeah, okay, no problem. Let's move to the next. It's sort of wobbly at this point of time. How you see your
okay.
custards and everything sort of wobbly, and it will take some time to not much time, maybe 5 minutes. But before that, I will get on to the next task, that is streaking bacterial cultures on agar plates. Now, can you shall we? Can you just okay? So this is something I had done for my experiment. I just wanted to show it off. This is the amount of
Wow.
agar plates that I poured. I'm going to use one of them. I'm going to make some streaks, right? All right,
Hmm.
right, great. Again, the most important thing, labeling what you are trying to do. You have to be very cautious about working. Overall, it just improves you as a person because you become more responsible when you're a researcher or a scientist. And
Absolutely.
today's date,
And supposing you're
yes,
working with many different bacteria, then you should know what you put on what, you know.
put on exactly. That's exact. Now, okay, can you see this?
Hmm.
Can you see this?
Yes,
Right? And
it's
this one,
solidified.
it's solidified, and this one is still a little wobbly, but then it is solidified. So yeah, our work is done. Now this has nutrient in solidified form, and I will grow bacteria on them. Now you all remember I spoke about bacterial stocks, right?
Yes,
That are stored at sub-zero temperatures. I'm going to remove one of them, and I'm going to streak them on these nutrient plates. So let's get started. Are you all excited for this?
yes,
All right.
very.
So we are going to make streaks using these micropipettes and tips.
So
I'll
let's
get this
one thing.
stock right now.
Let's just say that activity one is over. We made the
Yes,
plates,
Yes.
and now we are on activity two, which is
Yeah.
growing bacteria on these plates.
On these plates. Exactly. Thank you so much.
yes,
So we are done with this, and now we are moving to the next activity, that is growing microbes on a plate.
yes.
All right.
All right. So Ala has a very nice question. Ala asks, what will happen if we streak or grow bacteria on wobbly agar?
Oh, I mean, it will literally penetrate, and I can do that for you actually. Shall I do that? Shall I do that? I can, if I have any wobbly
Agar.
agar, I don't, unfortunately, but I will try to work on that, and I'll show you what happens.
So
Now,
it's
this
a
is,
nice idea, Nizam. Let's show them one with wobbly, as long as it
yes,
continues to stay wobbly, and one on solid.
yeah,
Yes,
because the thing is, this is solidified already, but let's see, because this is recently solidified. Let's see what happens if I am able to. Oh, can you see this?
Hmm.
Can you see this?
Not really clearly, but we get the gist. What is
Okay,
happening?
can you? It's not. It's making these
it's
dings
poking.
basically.
Oh,
It's
yeah, we
coming
can
off.
see.
Can you
Yeah,
see this?
Mm-hmm.
yes,
Mm-hmm.
Which is why you need to give more time for them to become solidified.
yes,
Can
yes,
you see
we
that?
can see
Okay.
Thank you
that.
for contributing to our show. That's your answer right there. Thanks for that question.
Yeah. That's amazing. Okay. Now, all right. Okay. So this is a non-wobbly agar pit that I'd made
Okay,
previously. And this is a stock culture that we are going to work with. Now, what I'm going to do is I'm going to take a sterile. Now this tip is sterile. This tip box is sterile. What I am going to do is I'm going to put some of them, some of the stock bacteria on the agar plate. Can you see the drop? I'm sure you might not because of the luminescence, but I'll show you what happens exactly, how streaks are basically made. So what I have done is I have put a spot on the agar plate, and I'm going to spread this.
Right.
Can you
So
see
this
the
one is not
action?
getting removed like the wobbly
Yeah,
one. Definitely.
absolutely. Right? So I'm going to spread it nicely, and then I will keep on diluting this, and you will understand why I dilute this when you see the result. Right? I'm making streaks at this point of time. I'm just making streaks in four
Yeah.
quadrants, in four different quadrants.
Yes. So what that basically means is just drawing lines from one end to another and trying to spread it all over the plate.
Exactly. Yeah. I'm going to show you what the result looks like because, again, it's a cookery show. So, yeah, okay. Can you see this?
Yes. So we can see lines on it now.
We can, yes.
Exactly. Yeah, yeah. So can you see also, and we will see later in the slide as well, there are certain colonies, spots, these are isolated colonies or isolated spots. Now, this is an individual organism, basically.
So
This
this,
is what happens after
Okay.
24 hours.
when did you streak this?
I streaked it yesterday.
Okay, you streaked
Okay. So it took 24 hours almost for it to grow this much.
Exactly, exactly, exactly. So this is what happens after 24 hours.
Hmm.
And I'm going to, so we are now done with the second activity, that is streaking bacterial cultures
Agar
Okay.
on an agar plate.
I
Now
think
let's
I have
move.
a few questions about this. Just you
Yes.
show up the bacteria, the show up the plate. You
Yes.
streaked it yesterday, and you grew it at what temperature?
I grew it at 37 degrees Celsius
One of those
because
temperatures.
because the organism that I have streaked, Staphylococcus aureus,
Hmm.
this is basically an organism that exists in human body. Now, again, I spoke about the optimal temperature, that is 37 degrees Celsius, which is why I grew it in 37 degrees Celsius.
You grew it overnight in an incubator at 37
Incubator.
Celsius,
Yes.
and the next day you actually see them that they've grown as these little colonies, what we call of structures.
Exactly.
Avisha asks, are these biofilms? And yeah, some people would define this as a biofilm.
Biofilm.
Collection
Yes,
of bacteria
absolutely.
on a plate is a biofilm. So they're like, I don't know. Yeah, I think Avisha, you should plan for Eurobiofilms 2024.
You should.
On that,
So,
next question is, Arav asks, can we show bacteria under microscope? We'll show you some pictures of that, Arav.
pictures of that, yeah, because it's very difficult for me to focus it on the microscope, but we have the pictures of it, and I'm sure you will get the idea of it. Do we have more questions? Or
Vedanshi asks, can
Yes, tell me.
microbes get injured from a pipette?
Can someone get injured from a pipette?
Can microbes get injured from a pipette?
They don't get injured by pipettes actually, because the size is so small. It's micrometers in size, and I don't think these do injure. They don't injure the microorganisms.
Great question.
That's an amazing question. That is all right.
So basically, the tip of the pipette is much larger than the microbe.
Exactly. So what I have streaked it, and I'm going to put it in an incubator. You all have seen the 37 degrees Celsius incubator, the huge cabinet, right? I'm going to put this for 24 hours, and we'll see what happens. And the result is basically this.
Yes.
Right?
Okay.
So this is the end of the second activity. Now let's move to the third activity. Now the third activity is basically taking these individual colonies and growing it in liquid cultures or liquid nutrient medium so that they grow and proliferate nicely. So I'm going to do that now. So now we move to the third activity. All right. Can you see this? This is a broth. This is a food for microorganisms.
This is the liquid one. This is not
This
going
is
to
a liquid.
solidify
This is
no
not going
matter
to
how long you keep it.
Exactly, because there is no agar in it. and I want to isolate these organisms. I want to grow these organisms into the liquid broth. Now, what I'm going to do is this is a lot of broth. I don't need that much. So again, we start all over again by labeling it.
So now you have tubes. You're working in tubes,
I'm working in tubes right
not
now,
plates.
not plates. Right. So I need to take some of the broth in these tubes.
Hmm.
I will take around 2 ml of the broth in the tubes. So this is a 1 ml micropipette, and this is a tips
Okay.
for 1 ml micropipette.
Okay. So in these micropipettes, how do you know that? How do you decide how much volume you're taking, or how do you
Yeah,
change that?
yeah, yeah. Okay, so that's a great question. So this is 1 ml, 1000
Yes.
microliter. You can change it like this. You can just rotate it.
Okay.
You see the numbers changing.
Yes, we can. We
Yeah,
can see the numbers are changing.
right.
So when it's 1000, that means 1 ml,
1 ml,
1
exactly.
milliliter. Okay.
1 milliliter. Yes. So now I'm going to add 2 ml in the tubes, 2 ml of nutrient medium, liquid medium in the tubes. Can you see the action of the pipette? There is some liquid in the tips of the micropipette.
Yes.
Right. Right. We have one tube. All right. We have the liquid cultures set with us now. This is where we will be growing our microorganisms. All right. Can you see this?
Yes. We can see a
That's
yellow colored
great.
liquid.
Yes, absolutely great. Absolutely.
Okay. So Arav has a
Okay.
question. Arav asks,
Yes.
can bacteria be grown both on solid and liquid?
Yeah, that's exactly what we are doing here. So what we did, in a recap, is basically we grew streaks on a solid medium, on a solid plate, and we're going to transfer these colonies. Can you see the individual
Yes.
colonies? Yeah, we're going to transfer this into the liquid broth.
Okay. So, Nizam, we have a really great observation that everybody made when you were putting the liquid in the tubes.
Yes.
So, everybody is asking, why did you change the pipette tips when you were putting liquid in different tubes?
Right. That's amazing. So I do this by practice. So whenever you want to work with two different microorganisms, right, you want to change the pipette tip. Now this came by practice. I need not do that at this point of time because it's basically the same liquid going in both the tubes. But normally when this has say one solution and this has another solution, right, so more often than not I would want to change the tips so that the contents are not mixed between the two.
Mixing, right?
Exactly.
Yes. And also sometimes when you're pipetting, if the tip touches something else on the outside, you don't know if that is
Yeah,
sterile as well, right? So
exactly.
you don't want to
Exactly.
mix things or
Yeah.
introduce random
Yeah. Exactly. That's
bacteria.
one of the reasons why I did this because it touched not it actually touched this part of the pipette tip box, which is why I was not sure whether it's going to be sterile or free of microorganism, which is why I changed the tip. Now,
Yes. So
okay,
all of this is part of good lab practice, right?
practices. Yes, it is called as good lab practices. All right. So what we are going to do is we're going to take a single colony. I've picked up one single colony. Can you see this?
Yes,
Yes.
It's creamish in
can.
color, right? And I'm going to release I'm going to release this in this medium, liquid medium. And next, this goes in an incubator at 37 degrees Celsius and under shaking condition. The reason is there will be more oxygenation when, more bubbling, more oxygenation when there is shaking condition introduced. So I will show you that right now. I'm going to place it in an incubator. So let's come with me.
Nizam, maybe we do it once more so they get the hang with the other tube. Just
Yes, absolutely. Let's do this. Yeah, absolutely. Yes.
Okay. So what you're basically doing is you're using the plastic pipette tip
Hmm.
and using that to pick up a colony of bacteria. So that's a bunch of bacteria. And then just putting that into the liquid tube, right?
Yes, that's exactly what I'm doing right now.
Okay.
Hmm.
So maybe once you take out the tube outside the hood, you can just hold it really close to the camera so everybody can see the tip inside it.
Yeah, we can sort of see it. Yes.
Yes, we
The
can see
others,
there's
Or
something inside.
we have more tubes, so we can show it to them actually.
sure,
We have the tubes which has microbes grown in
sure. Wow, wow. This is like seems like a sting operation.
it. All right. So what I'm going to do is I'm going to put these. I'm just going to get a beaker so that if
Yeah, so Arav, about your question, how to identify which bacteria grow, that's why you need to label the tube or the plate. Because the next day you really may not be able to distinguish it unless you have labeled it.
All right.
so can you
So
see them shaking now?
yes, we can
Yeah,
see them inside.
they're going to grow inside happily for 24 hours, and we will take it out later on.
Okay, so we again
Now,
leave them for 24 hours.
24 hours. Now I'm going to show you what what a grown culture looks like.
This is
And
basically
Okay.
yesterday, and this is to show them today.
yes, so this is can you see this? Okay,
Yeah.
this is just liquid medium without the bacteria.
Okay.
Can you
Okay.
see the intense? And now this is
Yes. Maybe
with the
Nizam, you can
bacteria.
just hold your glove behind it so that the light doesn't pass through and we can see the color
Yeah.
difference. Yes. Okay. So we can see one is very light and one is very like cloudy.
Yes. Let's maybe ask the young minds what difference they see. But
Yes.
Yeah.
what Snehal said is basically what we all observe. Let's see if they have
The
any.
second one looks jelly-ish.
Oh, yeah. That's a very good observation. It certainly is not, but then yes, it does look jellyfish.
Okay. Avishai says one is more brownish.
Color,
Yes.
cloudy,
It's cloudy. Yes, exactly.
like milky.
Yes,
Correct.
absolutely. All right. I have pictures of this, so you all can,
Okay.
make some observations there itself.
Yes.
Excellent.
Yeah. All right. Yeah. Okay. Now we
The
have
other
a
one
very
looks like something is added to it, which is the bacteria.
so we have a very small activity that is just observing them under microscope. Very small activity, not so much. But
Okay.
now what I have done is I have prepared a slide. Now this is a slide. This is a slide that is stained
Okay.
with a certain stain called as crystal violet. It will basically stain the microorganisms and it will render them visualized. So this is a microscope.
Okay.
I place this on the stage, right? I switch on the lamp and then a person will observe using a microscope. So this is a microscope that we were talking about.
Okay.
Right? And we will see what it looks like in the presentation.
Okay. All
All right.
right. So that's what a microscope looks like.
Looks like exactly.
Fantastic.
Okay. So So shall I go back to the presentation as well?
Yes, please. Yes, that would be
Okay.
great.
That was a lot of fun. That was a lot of
Yes,
fun. Yeah.
that was really fun.
Hello. Hi. Hello.
You're back. You're back. It's almost like there are two of you in the lab.
Yes, absolutely. Yeah. All right. So, the microscope does look cool. So, can you all type the three activities that we did? Only one word regarding the activities that we did.
Yes, that's a good idea.
Can yeah.
Very nice. Microbe
Yeah.
exploration, agar.
Agar. Yes, absolutely great.
Very
Yes.
Okay,
Streak.
streak,
Oh yeah.
perfect.
Okay. Yes, that's amazing.
Liquid
Liquid medium, micro visualization. Very good. Very
Wow.
good.
Okay.
Very good.
All right.
Bacteria
Okay.
culture. We've done a great job. I think we've done
Done
a
a
great
great
job.
job,
They've
Nizam.
understood it.
Great job. Great job.
Yes.
It's
Okay.
All right.
time.
Microscope.
We'll move
Yes.
on. But
Yeah, because we all running out of time. All
Yes.
right. So just the last bit, that is reading the results from a microbiological experiment. That is, you all could see that I was streaking them, and after 24 hours, it looks like this. It looks very fancy. You can see the isolated colonies. One colony, basically one microorganism will become one colony. It will populate, it will grow, and it will replicate and become one colony. So this is what the result of streaking on a solid medium looks like. And second is the observation that I was talking about. This is a liquid medium which doesn't have any microbe versus a liquid medium which does have microorganisms grown in it. And the third picture is basically Staphylococcus aureus, which is stained using a stain called as crystal violet, and hence the visualization is violet in color.
You see the beautiful violet. And you see them in
Great.
We do
clusters of grapes, right? Which is
clusters, yes, which
Yeah,
is
they look a little
yeah.
like
So
grapes.
exactly. So this is how we microbiologists make observations, because we know it that Staphylococcus aureus exist as clusters, like grape-like clusters. So we visualize it using that, and we can use these liquid growth for further experimentation.
sounds good. Okay. We
All
can
right. So
All right.
we can take questions now.
as well. Yeah.
Yes.
While they are
Sure.
typing their answers in the Google and Mentimeter, we'll take questions. Okay.
Yes,
Yes.
exactly. So thank you everybody for sticking through this amazing session with us.